We are in our lectures on synaptic plasticity, and we have discussed the different types of plasticity, mainly structural plasticity and synaptic plasticity, and the different types within synaptic plasticity, short term and long term plasticity. So, now we get into the side of modeling and using it in models for the effects of short term plasticity or those that are temporary. So, first of all, in order to see short term plasticity, we had said that if we measure the postsynaptic potential, let us say after we presynaptically produce a spike. So, this is I am drawing average diagrams here. We do this multiple times with enough gap and measure the postsynaptic potential, and we had said that there is some manipulation that is being done, and then we are measuring again. Actually, in the short term plasticity case, the manipulation is simply producing another spike, and what we can see is that the synaptic strength may reduce here, and then with another spike the synaptic strength may reduce even further, and with another spike the synaptic strength may reduce even further and be there, but after a delay of undisturbed period, we do the measurement again with a presynaptic spike. We measure the postsynaptic potential and we get back our original strength. So, this is the same as this. So, we have recovered our original strength, and in between we have what we call short term depression, that is, the synaptic strength is reducing with every subsequent spike. So, it is not necessary that it will happen all the time, but in general many synapses can show short term depression, where subsequent spikes produce short smaller sized EPSP, and there is a weakening of the synaptic strength. And obviously, this is also dependent on the gap between the spikes in the presynaptic side. So, if we have many closer spikes, then we can get even stronger effects of depression, or if we make the spikes presynaptic spikes much further, then we may even end up not seeing any change. So, it is dependent on how much presynaptic activity we are seeing. So, if this is the presynaptic activity, then that essentially is going to determine the properties of the short term depression that we are observing. And similarly, instead of short term depression, we can also see short term facilitation or potentiation, where we do have an action potential in the presynaptic side, and we measure the EPSP size. Let us say this is height of A, and the manipulation is producing another spike presynaptically, let us say within 50 milliseconds, and we see that the synaptic strength increases, and then with another spike we see that the synaptic strength increases further, and another spike we see increasing further, and later on if we allow the system to remain undisturbed and measure the synaptic strength again, we get back our original synaptic strength, where this particular EPSP is going to be the same as the last EPSP we are seeing, and hence regaining the original synaptic strength, that is by measuring the postsynaptic potential. So, how do these actually, I mean, why do these happen? Why is some synapse facilitatory and some synapse depressive in the short term? So, in order to understand that, we usually explain it based on the evidence so far, and with the idea that there is a limited amount of resource present in the presynaptic side, and that is the limited amount of neurotransmitters present in the presynaptic side, and that may be depleted and with successive action potentials presynaptically. So, this is how it may be thought of, that this is the presynaptic terminal. As you may recall, that there is a synaptic vesicle that is carrying the neurotransmitter here, and it is primed to be, or rather it is docked to be released in the synaptic cleft, that is the neurotransmitters will be released. So, when there is an action potential presynaptically, we know that these neurotransmitters are released into the synaptic cleft. Many of them go and bind to the receptors on the postsynaptic side and cause neurotransmission, and after that many are left to be taken back up, uh, endocytosed into the endosomes and then back into synaptic vesicles. So, actually the we can think of this as a cyclic process. We can say that the to make it a simple model first, we say that this pool of vesicles is the recovered pool. The pool of rather the pool of neurotransmitters in the vesicles inside the recovered pool, the ones that are effectively producing the postsynaptic potential, that is they are being bound to neurotransmitters, is called the effective pool. And the ones that are left in the synaptic cleft unused to be taken back up is called the ineffective pool. Or inactive pool, does not matter. And so, it is a three state process that is happening. So, we have neurotransmitter in this, which is made to be effective by going here, which goes to ineffective, and that is cycled back into the recovered pool. Now, the steps that are involved, this is they are unidirectional. We do not consider that the backwards flow can happen in this process, and the time constants of each of the steps play a crucial role in determining the entire the kinetics or even the short term plasticity. So, if we think of this as a three state model, that is we have the recovered pool, we have the effective pool, and we have the inactive pool. So, we have a time constant here, let us say tau R e. From E to I, we have a time constant tau E I, and from I to R, we have a time constant of tau I E, I R, sorry. Now, if we think of the way R is being or rather say R is changing, that is dr dt over time. If this is the concentration of R, so let us say normalized concentration, that is R plus E plus I is fixed to 1, that is the total concentration is 1 of the neurotransmitter, and R, E and I are basically the fractions, can be represented as normalized concentration. So, this is R dr dt. So, from I to R we have the process is driven by the concentration of I, and it is the time constant tau I R, and R is reducing due to the flow from R to E, which is driven by the concentration of R and the time constant tau R E. There is one missing aspect in this differential equation that we have written for dr dt. It is that all the things are not happening continuously. So, R to E is only happening when there is an action potential. So, when there is an action potential, only then the vesicles in the inside of the presynaptic side is going to release the neurotransmitter. So, this rate of reduction in R must be happening only at the time points of spikes, which may be indicated by U, where this U is generally 0 and will be considered to be 1 for a brief 1 millisecond period or some short delta time period, and then it will go back to 0. That will essentially represent that the depolarization has happened and the calcium comes in, and the recovered pool sends neurotransmitters into the effective pool. So, that is what we are representing by this U, which can be taken as 1 for the spikes for that small duration. So, now if I have the d E dt, that is basically being increased due to the flow from the spiking, that is U times R by tau R E, and is being reduced due to the flow from E to I, and that is basically the concentration of E divided by tau E I. So, all are first order kinetics, and using this equation we can also write from the diagram itself, that is d I dt is simply increasing due to E by tau E I, and it is reducing due to the I itself going to R, that is tau I R. So, now with this model, if the tau R E is of the order of 1 millisecond, this practically is of the order of 100 milliseconds, and this of the order of 1000 or greater than 1000 milliseconds, that is more than a second in multiple seconds, that is the time constant. So, this becomes the rate limiting step in the whole process. That is, if we have an action potential, that is this U becomes 1, then R is reduced, and that triggers the flow in the forward direction from R to E to I to R. So, initially all the neurotransmitter is in R, where R is equal to 1, and after that, with every spike we are that R is reducing and going into E and I. However, the rate of change of I, that is going from I to R, is extremely small, because this tau IR is very large, and that basically keeps the neurotransmitter in the ineffective to the R pathway in the middle, in that state. So, it is in I and trying to move on to R. So, very little is actually being going to the recovered pool. So, the reduction in the neurotransmitter is a reason for the synaptic depression to happen when we have many spikes simultaneously in a closed time period. So, for every spike we get some reduction in the postsynaptic potential, and we can actually show with this model by simulating it that there is synaptic depression. So, this synaptic depression model works very well and has been used in multiple models with this 3-state model. So, in fact, with this idea, we will be discussing one of the consequences of short term depression later on. So, now there may I mean, this is only a simple model. Now, there are many other parts of this short term depression, and that has to do with the calcium that is coming in. If you recall, for this U becoming 1, we actually, it is based on the calcium coming in due to the spiking. So, this entire model can be further modified by qualifying that U and bringing in the calcium concentration explicitly, instead of making a 0-1 kind of transfer from recovery pool to the effective pool. So, in order to do that, I mean, the reason why we will do that is actually this model that we have talked about does not explain short term facilitation, because this only can explain short term depression based on the time constants that we have discussed. And so, what can we do? I mean, basically there is always a probability associated with the release, that is, the synaptic transmission is also probabilistic, and so overall neurotransmitter available may be NT, but it is now being multiplied by a probability in order to be able to bring in the effect of the calcium, because the calcium is effectively what decides the probability of release. So, actually when we have a lot of activity like this, in small synapses there is a build up of calcium, that is the residual calcium hypothesis, where due to every action potential the calcium is building up, only some of the calcium is used up in releasing the vesicles, releasing the neurotransmitter in vesicles, whereas in the next action potential, due to the remaining calcium and more calcium added up, the calcium concentration increases, and hence the probability of release increases, and there is an increased number of neurotransmitter released, and effectively we have a larger postsynaptic potential. So, if we want to go ahead and include and actually show facilitation, we have to go and explicitly modify that u and bring in this probability term, and there will be further other things that need to be included. There is one more aspect here where we can have this R may be broken up into two pools, and that has its own consequences as well, where R is one is the recovered pool, and then we can have another state that is a readily releasable pool, that is the ones that are only readily releasable, that goes into the effective state. So, we may be, there is no direct path per se as we have had in the model that we have described, but there can be an additional stop, which is the readily releasable pool, and that we will discuss along with the inclusion of the probability term and calcium concentration in our next lecture on short term depression, short term plasticity. Thank you.